Last unit, nucleic acids. We know these as DNA and RNA. It's recognized, okay, that the physical traits are inherited as genes, and the genes are located on chromosomes. And when you're talking about a complete set, the entire set of genetic information, we refer to that as the human genome. And you'll hear me use that term frequently. We have diploid and haploid cells, haploids, which are the gametes of sex cells. All this stuff should be familiar to you from your biology class. Molecular biology is the study of DNA and RNA nucleic acids. Now, we're on the last of our biological macromolecules, the nucleic acids. Their monomeric unit is a nucleotide, and there are structural features that can be identified that will allow you to differentiate nucleic acids from the other three classes of biological macromolecules. Let's see what makes up a nucleotide. First of all, we have a nitrogenous base. We'll see what those are. Those are purines and our pyrimidines. We have a sugar, which, as it turns out, is a ribose unit. And lastly, we have a phosphoric acid residue. These three components are necessary to form the monomeric unit of DNA and RNA. The nitrogenous bases, the order of the nitrogenous bases is what specifies the genetic code. That's what is interpreted off the messenger RNA, ultimately translated into proteins, which express everything that we are. Okay? Everything, our height, eye color, hair color, everything. We have two types of nitrogenous bases, the purines and the pyrimidines. The purines, there's adenine and guanine, which we usually abbreviate with a single capital letter, the beginning of each name. So adenine is A, guanine is G. The pyrimidines, we have C, T, and U, cytosine, thymine, and uracil. We'll see how those come into play in terms of Watson-Crick base pairing rules in a little bit later. These are the structures of the purines and the pyrimidines. No, you don't have to memorize them. Okay, I'm just giving this to you just so you have it for future use. You can see the purines are two fused rings, whereas the pyrimidines are a single one. Interestingly enough, thymine and uracil, they're very, very similar. Look at the structures. The only difference here is at C5. Okay, in thymine there's a CH3. In uracil there is a hydrogen. And we'll see that thymine and uracil, they replace one another. Uracil is only found in RNA, whereas thymine is only found in DNA. This is an actual nucleotide. Here's our nitrogenous base. Our nitrogenous base is connected to a ribose. Now, a ribose is a 5-carbon sugar. Notice here, oxygen, one to the right, the anomeric carbon, right? Carbon number 1, 2, 3, 4, 5. We put these little primes on here to differentiate the numbering system from other numbering systems that might be used to number other things in other areas of the molecule. So we'll be referring to 1 prime, 2 prime, 3 prime. But just know that normally in our chapter on carbohydrates, where we numbered our carbons 1 through 6 or 1 through 5, these are the same numbers that correspond to that. Notice it is going up, right, which makes it beta, right, beta. At the 5 prime carbon is where we have the phosphoric acid residue attached. This is a nucleotide. This is adenine. Adenine, when it's attached, actually becomes adenosine. So there are subtle little differences in the name that allow you to determine what its current configuration is. Notice here, these are virtually identical, all right, these two nucleotides, except for carbon number 2 of the ribose ring. In one, there's no oxygen, and in the other, there is an oxygen. Everybody see that? Without the oxygen, this is deoxy. Deoxyadenosine 5' monophosphate. You're not going to have to learn the names or name them using this terminology. But know that when there's no oxygen there, it's deoxy. That's DNA, deoxyribonucleic acid, versus RNA, which is ribonucleic acid. And it's interesting. That's the only difference, right? Well, that and the fact that uracil is an RNA, and thymines and DNA. But the absence or presence of oxygen at the 2' carbon of the 5-carbon ribose ring determines whether or not you have a nucleotide for DNA or a nucleotide for RNA. Now, the numbering of the ring, the ribose ring, gives that nucleotide directionality. And we'll see that it's actually, when we start attaching these monomeric units together, forming a segment of DNA or RNA, that numbering that chain gives that strand directionality. And we're going to see that the two strands of DNA actually run in opposite directions. Again, this is just the same slide, only the units are put in here. Here's our ribose unit, phosphoric acid, 5'. The 3' OH, this is going to be important. Okay, it turns out that this is where we're going to run our linkages when we connect adjacent nucleotides. Our bonds are going to be at the 3' OH and the 5' phosphate. But we see here the difference. It's just labeled for you so you can see them more clearly. Now, if we look at a linkage, okay, we have a phospho-sugar, 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 phospho-sugar. This is the backbone, and what we refer to as the backbone of DNA or RNA. And we have the nitrogenous bases actually hooked here at the anomeric carbons. Now, if we look closely at the linkage here, notice that it's the 5' phosphate that's linking to the 3' OH group. We call this a 3' to 5' phosphodiester linkage. Now, remember what an ester looked like in organic chemistry. We had a C double bond O, single bond to an O. That's our ester linkages, remember? C double bond O, single bond O. This is an inorganic ester because it doesn't have carbon in the center. It has phosphorus. And you see phosphorus, double bond O, and then single bond O. That's one ester linkage. And then it's got a single bond O on the other side. That's where the diester comes from. It's the phosphodiester linkage. Okay? Now, here's a little segment of a nucleic acid. And no matter how many of these you put together, whenever it stops, you're going to have one end where the 5' phosphate is open. And you're going to have another end, which we can't see here, that has a 3' OH. It's very similar to what we saw with the assembly of the amino acids and the production of proteins. No matter when we stopped, we were going to have an amino terminal and a carboxyl terminal. The same thing is true here. And so whenever you have a segment of a nucleic acid, you will have a 5' end and a 3' end. Okay? 5', 3'. Everybody see that? Again, note the glycosidic bond linkages here. Beta. Does everybody see that? 5', 3'. So if we were to look at, represent this segment of DNA, we would take a quick look at it. We would say 5 prime, we always write DNA in the 5 prime to 3 prime direction. So we'd have 5 prime A, G, T, C, 3 prime. So the previous segment, or that segment, this is what it would look like. And there are many different ways to do it. Sometimes books put a little P here for phospho, but the easiest way is 5 prime to 3 prime, A, G, T, C. Everybody see that? Now, DNA is a double-stranded helix, a double helix, which has 2 strands running in opposite directions, which we call anti-parallel. Right. So let's see the H bonding between the nitrogenous bases. This is called base pairing rules. Here is a segment of the DNA. And the best analogy I can give you is to think of a rope ladder. Okay? You take a rope ladder and you connect the rope ladder at the top or bottom, it doesn't matter, at the top, and then you wind the rope ladder around the central axis. You don't twist it. You don't take the rope ladder and twist it. You hook it up and you wind it around. That's the best picture of a segment of DNA. You're connected at the top, and this rope ladder is wound about this central axis point. So you can see the ladder is spinning around it. It's not twisted at all. Okay? Notice 5 prime, 3 prime. The other strand is 3 prime to 5 prime. Anti-parallel strands. Look at the backbone. It's negatively charged because of the phosphate group. See all the negative signs on there? And that's actually the reason why gel electrophoresis works. Okay, this is how come we can fragment or digest DNA and get the segments of DNA to move through a gel. Push electricity through. It's got a negative backbone. It's also why in order to stabilize DNA, you need cations. Okay, we call them chelating agents. They're positively charged, usually divalent, like magnesium 2+. These are instrumental if you want to get strands of DNA together. Okay, does everybody see that? And you can see these. Look at these 2 crevices right here. We have a wide groove and a thin groove. A wide groove, thin groove. They're investigating that today in terms of medicinal purposes, maybe activating certain segments of DNA, the genes. You notice here that the strands of the ladder are held together by the nitrogenous bases interacting between each anti-parallel strand. And that's what we're going to talk about next. Watson and Crick's base pairing rules. Discovered in 1953. Really, probably the single most significant discovery in the 20th century. Some people would argue the 21st century. The base pairing rules are what allowed us to determine what the structure of DNA was. And of course, that was a landmark discovery. They are well defined. Adenine always binds with thymine. Guanine always binds to cytosine. Just remember at, okay? They are held together by hydrogen bonding. We'll see that in just a minute. G always binds to C. So if we were asked to write the complementary strand of DNA, which we call cDNA, to this particular segment, what we would have to do first is we would run the segment 3' here, and then we'll employ Watson and Crick's base pairing rules to determine the segment, and then we would end with 5'. But we always write DNA in the 5' to 3' direction. So we'd have to pick this up and flip it over. in order to get the complementary strand. It's not enough to simply write the base pairing. Note, every place I have an A, I have a T. And when I have a G, I have a C. They're all base paired together, according to Watson and Crick. Everybody see that? But I'm not done because I was asked to write the complementary strand, so I have to flip that around. I move the 5' over here, I go G, C, I'll do it quicker if I go G, C, A, T, A. We have to do it that way. Okay, we have to write in the 5' and 3' direction because we're going to see the codon tables for the translation of messenger RNA is in the 5', 3' direction. So we have to do it this way. So we might as well just get in the habit right now. Everybody see that? Understand? Watson and Crick? I'm reasonably sure somewhere on some exam or lab quiz or lab something, you're going to have to write a complementary strand. For sure. Everybody good with that? See you feverishly writing it down. What are you doing, writing down the sequence? Got it? You can base pair it later. You can pair that later. Okay. Here's the hydrogen bonding. Notice that we always have a purine with a pyrimidine, right? And the hydrogen bonding, remember, A always goes with T. At is a two-letter word, so there's two hydrogen bonds that hold the A's to the T's. Okay? GC actually has three distinct regions of hydrogen bonding. And the significance there is that if you were to see a heavy concentration of GCs in a region on the DNA, then you would say, well, that region's more tightly bound than if they had ATs. But there's the hydrogen bonding for that. Here's an actual segment, okay? We have the 5' end here. We have the phosphate sugar, phospho sugar, phospho sugar. The phosphodiester linkages are linking 3' OH to 5' carbons, okay? We look here, we're running the anti-parallel strand, so the 5' end is down here. Phosphodiester linkages on the phospho sugar backbone, and then we have our nitrogenous bases base paired here. We have T with A, we have T with A, we have C with G. So this is what an actual double helix or double-stranded DNA would look like. Okay, no, again here. 5' to 3' directions in this way. Everything's 5' to 3'. So when we think of, for example, our carbohydrates, we think of alpha 1,4 or beta 1,4 linking our monosaccharides. When we think of lipids, we think of esters. The esters linking the fatty acids to the glycerol molecule. When we get to proteins, we think of amides, okay, the amide linkage. When we talk about nucleic acids, we think of phosphodiester linkages. Okay. At minimum, these are the things that you absolutely have to know. What linkages are responsible for connecting the monomeric units, in each case, connecting them together to generate these large biological macromolecules, these polymers. This is just another way of seeing it. There's our 5' end, got a little color coded here. Our nitrogenous bases are these orange, and our sugar units are blue, and we can see our phospho-sugar backbone here in this kind of green color. So you see it is just like a rope ladder, where the rope on each side are the anti-parallel strands of the phospho-sugar backbone, and the rungs of the ladder actually represent the base pairing rules of the nitrogenous bases. Okay, where each strand contributes half a rung, and you get the full rung. And then they're winding around a central axis. Here's a space filling, and again, you can see these wide grooves and thin grooves. And if you think about a rope ladder being wound around a central axis like this, for example, if we connected it here and we wound it around without twisting it, we're going to have two grooves. One of the grooves is going to be set by the thickness dictated by the rungs of the ladder, and the other one is going to be set by how far apart the winds are. Right? So we have a wide groove and a thin groove, and it's not differentiated which is which. But it is believed, I mean, currently they're doing research on trying to activate, put molecules or atoms into these grooves in an effort to try and activate certain areas of the nucleic acid to maybe produce, certain types of proteins. It's current research. Pretty good stuff, too. Eukaryotic DNA. 3 billion base pairs. 3 billion. Now, they just completed the Human Genome Project where they sequenced the human genome. I think that was completed in the early to mid-90s, don't quote me on that. But it took an awful lot of laboratories an awful long time to sequence the human genome. And we were alive when it was done, so that's a novel discovery in molecular biology and biochemistry. I like this. DNA is the largest linear covalent molecule in nature. If it were roughly the size of a strand of spaghetti, it would be over 10 miles long. That's pretty long. Largest linear covalent molecule in nature. Kind of cool. Here it is. Here's the individual DNA, which we call the naked DNA. You wrap around these histone proteins, which have a high concentration of basic residues, which are positively charged. There's no surprise there that the histones will have some positive charged residues in it because the backbone of the DNA is negative. And there's a whole folding process that goes on here with the histone first folding around the histones and creating this beads on a string, and then those stack and those further stack and stack and stack until eventually they compress down into the chromatin, which makes up the chromosome. Very efficient, very, very efficient in the packing of DNA. DNA replication is mediated by a variety of enzymes. We don't want to see this particular text because this isn't our text. 4 major differences between DNA and RNA. First of all, RNA contains ribose instead of deoxyribose. Okay, we saw that difference. That's nothing more than the carbon number 2 of the ribose ring does not have oxygen for deoxy, does have oxygen for ribose. RNA contains U instead of T, contains uracil instead of thymine. RNA is single-stranded and therefore much smaller than DNA. Some viruses, there are exceptions. And of course, right along with that single-stranded, RNA is much, much smaller than DNA. Much smaller. These are the major differences. Protein synthesis. How do we get our proteins from our DNA? Well, the information is encoded in the nitrogenous base sequence in the DNA. So how do we extract that? How do we specify, or this is specifying the amino acid sequence of the proteins, but DNA and RNA have monomeric units that are nucleic acids. Proteins have monomeric units that are amino acids. These are two entirely different languages. So how do we get from one to the other? Well, the central dogma of molecular biology is, and I had to look up dogma, because I didn't know what it meant. Something held as an established opinion. Okay? Is that you have DNA, and since DNA and RNA are both made up of nucleic acids or nucleotides, now I know there's a subtle difference. One doesn't have oxygen on carbon number 2 and the other does, but they're both made up of nucleotides as their monomeric unit. So this is the same language. So we transcribe. Transcription is what does that. Okay, we basically copy. When we go from RNA, which is in nucleotides, into proteins, which is amino acids, this would be a different language, so we translate. Okay, so DNA is transcribed into RNA, RNA is translated into proteins. And a good way to remember that is just how I said it. These are both nucleotides, so they're scribed, transcribed. This is nucleotide, amino acid, translation. Different languages. So, there are three types of three types of RNA. And again, unlike DNA, RNA is single-stranded. It's single-stranded and much smaller. First, we have messenger RNA, which is abbreviated lowercase m RNA. This is the template for protein translation. It is the carrier of the genetic information. Transfer RNA, abbreviated lowercase t RNA, this is what actually transports the amino acid at the direction of the messenger RNA. And we'll see the transfer RNA actually has an anticodon, which binds to the codon, which is a sequence of 3 nitrogenous bases on the messenger RNA. Lastly, ribosomal RNA. This is a major component of ribosomes. It is the most abundant form of RNA in living cells, approximately 80% of all RNA. Now, the ribosomes are the site where protein translation takes place. let's do transcription. Okay, remember, there are no thymines in RNA. Now, if you have another way to do it, that's fine. If you understand the whole AT and AU pairing and whatever, that's cool. I don't have a problem with that. But I always get confused with it. So what I do is I follow the same process as if that I use to generate a complementary strand of DNA. So here's my segment, okay? I follow Watson and Crick base pairing rules, and I put my complementary strand, right? I flip it. I C, G, T, T, A. I flip it, put it in the right direction. Then, wherever there's a T on here, I simply substitute in a U. And that's how I get my messenger RNA. That's how I do it. That's how I do it. Other people, much smarter than me, can look and say, well, look, if I have a T, I still pair it with an A, but I'm dealing with messenger RNA. So anytime I have an A, it's actually going to pair with a U. So these two ultimately work right here. So whatever way you want. I draw my complementary strand, flip it around in the right direction, then I just draw a line through all my T's and put U's in. Everybody good? Translation. This occurs at the ribosomes. There's two subunits, a small ribosomal subunit and a large ribosomal subunit. Let's see where we're at. Okay. We have, let's go back. In the cell, the DNA is located, in a eukaryotic cell anyway, the DNA is located in the nucleus. And the nucleus is bound by the nuclear membrane. Okay, so within the cell itself, you have a nucleus. And the nucleus is surrounded by its own separate membrane. But the membrane has these tiny little nuclear pores in it. Okay? So the RNA is transcribed in the nucleus. And the RNA, because it's smaller and single-stranded, can wiggle through the nuclear pores out into the cytoplasm. Okay? Cytoplasm is basically everything outside the nucleus, but inside the cell membrane. Once the messenger RNA is out into the cytoplasm, it's met by the ribosomal subunits. First, the small ribosomal subunit will bind at a recognition site on the bottom. And then the large ribosomal subunit will come down and sit on top of it. First, the bottom one will bind, and then the large one will sit down on top. And it will start translating the messenger RNA to produce the protein. That's basically what's going to happen. We'll see more of that right now. Now, it's actually the base pairing between the codon, and now a codon is a set of 3 nitrogenous bases, which is read, it's called a reading frame, on the messenger RNA. So you have this long segment of messenger RNA, and it's read 3 nitrogenous bases at a time, which we call a codon. Now, does anybody have a guess why the codon contains 3 nitrogenous bases and not 2 nitrogenous bases? Why do you think the codon would have to be 3 bases? Well, how many bases do we have? In RNA, right, there are no T's. So it's AU and GC, right? There's 4. If you have 4 things, say you have 4 different colored balls, and you wanted to show a description using only 2 of them at a time, how many different combinations do you think you have? Well, statistics, which we don't care about, there's 16 different combinations. So if you have 4 nitrogenous bases and you want to represent a description of those 4, but you only want to use 2 of them at a time, there's only a total of 16 possible combinations. Does anyone see a problem with that? What's the problem?
Well, there's not enough variation
To account for how many amino acids do we have? 20. We have more than 16, right? So if you wanted to use a codon that only represented 2 nitrogenous bases, you would not have enough combinations to be able to explicitly identify individually each amino acid. There's not enough combinations. But if you go to 3, you have 64 possible combinations. More than enough to differentiate each individual amino acid. In fact, now you can actually have multiple codons to specify a single amino acid if that amino acid is, say, more important than the others. And there are. Some amino acids have as many as 6 codons, where the others only have 1. And if you have 6 codons, that's great. That means you can tolerate mutations in the sequence, but still end up with the same amino acid. Right? So there's our codons. The tRNA will bind at the specific 3-base sequence that's complementary to the codon, and we call it the anticodon. So the transfer RNA has the anticodon. And again, we just call it anticodon because it's complementary to what we're defining as a codon. This is a codon table. There's a question I asked you right there. This is a codon. Here's the first nitrogenous base, second base, third base. Okay? Down here we have home run. No, just kidding. First base, second base, third base. So if we have, let's look at leucine. If your first nitrogenous base is C and your second one is U, it doesn't matter what your third one is, you're going to get a leucine. Okay? You're going to get a leucine. In fact, right here, leucine has 6. Do you think leucine is an important amino acid? Absolutely it is. If it's got 6 codons, your genetic code, your DNA, can withstand several point mutations that changes a specific nitrogenous base, but still encodes for the same amino acid. Okay? That's why protein sequencing, the primary sequence of proteins, is not as accurate as running DNA electrophoresis. Okay? Because you could have similar proteins because the amino acid sequences are the same. But once you dive into the actual nitrogenous base sequence, the codon, that's where you'll find the variations. Okay? It's one where you have the expression of the amino acid versus the coding for the amino acid. Notice there's only one start sequence, AUG. That's methionine or start. We have 3 stop sequences. To me, that's kind of unusual. You'd think it'd be the other way around. You have more starts than stops, because let's say you want a tyrosine, right? And you have a point mutation that changes a C to an A. And instead of UAC and putting tyrosine in the middle of your protein, or wherever it goes, you hit a UAA and hit a premature stop. Protein translation stops. The protein is now functional. Premature stops are almost 100% denatured. The protein does not function at all when you hit a premature stop. Now, this is a messenger RNA codon table in the 5' 3' direction. That's why you have to make sure and switch. Okay? If we back up, for example, here's our, let's say this is our cDNA in the 5' 3'. And when we change our T's to U, this is what we get. CGU is our first amino acid. Now, actually, it wouldn't be. It'd have to be AUG, right? Because methionine is the start sequence. It's the recognition sequence where the small ribosomal subunit binds. But just for the case of argument, let's say that we didn't. We kept it in the 3' to 5' direction. If we just change these to U's, these T's right here to U, our first one would be CGA instead of CGU. Now, I don't know if that's the same amino acid. It probably is, because we're videotaping these. But let's see, CGA versus CGU. CG, yeah, A versus U, it would be the same, arginine. But if you go down that sequence, you'll find something different. Okay, so that's why everything's 5' to 3'. Arginine looks like another one, right? As long as you have CG first, it doesn't matter what the last one is. And there's 2 more here. Okay? Okay, so what happens? Again, our messenger RNA is transcribed in the nucleus. It weasels through the nuclear pores, gets out into the cytoplasm. And out in the cytoplasm, the small ribosomal subunit recognizes the initiator sequence. Notice here, it's AUG. That's our start sequence. The transfer RNA with the anticodon UAC, which is anti to AUG, sits down in the binding site carrying the methionine amino acid. The large ribosomal subunit sits down on top of the messenger RNA and sandwiches the messenger RNA between the 2 subunits, the ribosomal subunits. There's 2, a small and a large. Now, the small and large ribosomal subunit are only together during active protein translation. All other times they're apart, floating around. Once they sit down, once the large one sits down, you define 2 sites, the P site and the A site. Now, I think of the P site as being the peptide site. It's the one that's got the growing peptide. But it's got a name, peptidyl tRNA binding site. The codon right next to it is called the A site, which I think of as amino acid. That's where the amino acid that's going to add to the chain is coming in. It's also got a name called the amino acyl tRNA binding site. So, we've defined our P and our A site. Our peptide, or our growing peptide chain, is located in the P site. The next reading frame is in the A site. That codon is being translated, and the tRNA possessing the anticodon that's complementary to the codon sits down in the A site. The entire peptide is transferred to the incoming amino acid. So it all ends up on that initial or incoming transfer RNA. The whole ribosomal subunits shift up one reading frame, which is one codon. What used to be the A site becomes the P site, and the amino or transfer RNA that was in the P site is released to go back to the amino acid pool to pick up whatever amino acid corresponds to its codon. And this keeps going, okay, translating one at a time, one codon at a time, until you hit the stop sequence, until you hit the top codon. Now, as these amino acids are being assembled, the complex interactions are occurring as it's being assembled, one at a time. They're the hydrogen bonding and the hydrophobic interactions and all of those covalent and non-covalent interactions that we talked about in protein structure. That's all happening right here. Okay? It's all happening.